Good day, everyone. Welcome to our Tuesday get-togethers on the cerebellum. It's good to have you here with us. I am so pleased to have with us today Jason Christie. Jason studies synaptic physiology at the Oregon Health Science Institute with Gary Westbrook, then did a postdoc with Craig Jahr. He then was a group leader at the Max Planck Institute in Neuroscience in Florida, and now he's a professor at the Department of Physiology at the University of Colorado. His latest work on the molecular layer in neurons are particularly interesting and thought-provoking, and I'm so happy that he's going to be describing some of that work for us today. Thank you for being with us, Jason.
Thanks, Reza. It's good to participate in this format again. It's grown a lot since I think the first time I went, which was one of the first ever. So it's great to see the growth of this and the exchange of within the cerebellar community. So I'm grateful that you're supporting this. In contrast to the last talk, which is more about online control, the cerebellum has on behavior. This is going to focus on our interest in neural circuits for cerebellar learning. It's motivated by, I think, our longstanding interest in algorithms underlying fine motor control. Specifically, how do we generate these wonderful feats of movement, such as this gymnast performing a backflip on a balance beam, although these things generally apply to more mundane tasks like reaching for a glass of water or even having well-calibrated eye movements. Notably, these are, our ability to perform movements carefully. It's not innate. Rather, it's learned through practice, often through trial and error, where we make mistakes and learn from those mistakes. So the brain is believed to implement supervised motor learning algorithms where a motor plan is sent from the brain to the body for execution. And the resulting outcome can be compared to what was intended, and that there's a deviation. This can trigger adaptive alterations in response to those motor errors and can be stored as memories that are recalled on subsequent trials to increase or optimize the performance. So the cerebellum is a well-known site for supervised motor learning. It can work as an adaptive element on motor programs to alter them and improve performance. But notably, that has to be trained. And so it's widely understood that there's feedback signals that can get sent to the cerebellum that instantiate plasticity and alter output on subsequent trials in response to that mistake. So this pathway into the cerebellum is fairly well understood to be delivered by climbing fibers emanating out of the inferior olive, and climbing fibers are well suited to produce these update signals that generate instructions for adaptive updating based on their unique connectivity and physiological responses that they produce. So each Purkinje cell, the primary output neuron of the cerebellar cortex, receives an input from a single climbing fiber, but that climbing fiber makes several hundred very powerful excitatory synapses, such when the climbing fiber fires, it absolutely produces a explosive response in the Purkinje cell. That is, the resulting synaptic depolarization can enter the axon initial segment and trigger a burst of simple spikes that then are partially back propagated into the soma and are registered as a Purkinje cell complex spike. And so these are the well understood neurophysiological signatures. Notably, the complex spike is really an output. It's generated in the axon and sent down the axon, and you just see a partial reflection of that waveform in the soma. Importantly, for instructional updating, that rather large synaptic depolarization can enter the distal dendrites of the Purkinje cell, which is a highly electrogenic region, and initiate a burst of calcium dendritic action potentials, as shown here in this simultaneous recording that Matt Rowan, a former postdoc in the lab, performed a number of years back. So this is the comparison of the signature both at the dendrite and the soma, and you can see the waveforms are quite different because the underlying ionic conductances supporting each are separate from each other. So these dendritic calcium action potentials can then propagate throughout the dendritic arbor, elevating intracellular calcium. And calcium is a near universal signaling molecule for plasticity induction. Hence, the resulting climbing fiber evoked calcium response in the Purkinje cell dendrite is notably critical then for instructive updating. And what's great about these calcium signatures is that we can record them using calcium imaging technologies. For example, we can express the calcium sensor GCamp in Purkinje cells and measure their activity in vivo. This is just an example where Mike Gaffield, another postdoc in the lab a few years back, recorded activity in a quiescent animal. Here we're using 2-photon microscopy to image dendrites of GCamp expressing Purkinje cells in the lateral cerebellum. And you can see in these representative Purkinje cell dendrites, this ongoing dendritic calcium response. Each calcium event is demarcated below. It's detected by an algorithm. And each of these is associated with a complex spike. So we know that they are climbing fiber mediated. And Mike went on to suppress the output of the inferior olive, and this calcium activity goes away, indicating that substantial amounts of Purkinje cell calcium is largely mediated by climbing fiber directed input. So apart from showing an interesting set of calcium technology to record activity, this also shows an important feature of climbing fiber signaling that we're all generally aware of, is that climbing fibers fire regularly, essentially at 1 to 2 hertz, and that's ongoing. So this is just a quiescent mouse doing anything, and you can see it's just continuously bombarded with excitation. This continual bombardment offers potential problems. Modeling suggests that if all of these climbing fibers were super potent for plasticity induction, this would be potentially problematical because it would potentially lead to inappropriate associations and potentially maladaptive behavior. So we've long thought that that there might be a regulatory mechanism that, in addition to climbing fiber signaling, regulates their ability to produce plasticity in certain contexts. For example, during correct performance, you might want to have a system that regulates the potency of these climbing fiber signals such that they don't evoke inappropriate alterations to an already well-performed behavior. So to explore this, we've been using one particular task or behavior that is looking at the vestibular ocular reflex and its adaptation. It's great because we can carefully control the context that the mouse exists in to produce adaptation in a very directed way. For those that aren't very familiar, the VOR is an opposite direction eye movement to head rotation. We can elicit this in an animal by head fixing it on a platform and rotating that platform so we can measure the displacement of the eye. The VOR is accomplished by a 3-synapse loop in the brainstem, where it takes these vestibular information and the brain converts it into eye movement coordinates that move again in the opposite direction of the head. And this enables us to maintain stable gaze. In fact, if we move our heads and our eyes don't move in accordance, we will experience visual motion, and the brain uses the feedback of visual motion to gauge how well that behavior is performed. So the presence of visual motion during the VOR indicates an underperforming VOR. If visual motion or retinal slip occurs in the opposite direction of the eye movement, this indicates an underperforming VOR that requires an adaptive increase in its gain to regain accurate performance. And that's illustrated here. So if you have visual motion, this is represented as this is the feedback that drives adaptive learning. And a number of labs have shown that this actually induces activity of climbing fibers that can then produce learning. So the feedback signal from this visual motion is fed to the cerebellum, specifically the specialized region of the vestibular cerebellum called the flocculus. This is where learning is instantiated to recalibrate that VOR in response to an underperforming VOR. And therefore we can use this behavior to gauge what's happening in the flocculus to generate those learned changes in behavior. Provided that we're interested in climbing fiber evoked signaling in Purkinje cells, we could first turn to electrophysiology to record climbing fiber induced activity in Purkinje cells. Here's an example where we inserted a silicon probe into the flocculus and then identified Purkinje cell units, those that include complex spikes. And then we could introduce an adaptive stimulus to the animal that is rotating it on this platform and then introducing visual motion that moves in the opposite direction of the head. And so this basically mimics the animal making a mistake. It actually tricks it to believing that it does make a mistake. If we repeatedly pair this over tens of minutes, what you can see is there is an adaptive increase in the gain of the VOR. That is, there's an increase in the amplitude or the size of that eye displacement relative to the head. Typically, it's 15 to 20%, and it's quite robust across animals. So this is a very potent adaptive stimulus that we can record this behavioral change. So the question is, what's happening to climbing fiber evoked activity in Purkinje cells during this stimulus? And by examining that activity during the silicone probe recordings, you can see relative to the VOR performed in the darkness to this gain increased stimulus. This again, the stimulus that will produce a increase in the performance of the VOR. You can record a deep modulation of climbing fiber evoked activity in the Purkinje cells registered by an increase in the rate of complex spikes. And so this graph shows a summary across Purkinje cells. There are Purkinje cells that we can see with increases in both phases of the vestibular stimulation, so for both leftward and rightward head turns. So that would be ipsiversive and contraversive to the site of recording. So this fits with engagement of the idea that there's an engagement of climbing fiber evoked signaling during this erroneous context, as you would generally expect based on prior results. However, what's great about this behavior is that we could also present a context where the animal's behavior is already well calibrated. And that's similar to a context that we exist in. That is, if the head is turned, when we turn our heads and we move our eyes in the opposite direction, there's no visual motion. So there's really no feedback signal that would generate an adaptive change. And so in this context, what you'd like is potentially regulation over any induced climbing fiber activity, else you would degrade an already well-calibrated response. And so we could elicit this in a mouse and similar to what we had done before. Again, rotate this mouse on a platform, and then instead of moving a visual stimulus in the opposite direction of the head, we actually just keep it still. And so the animal's experiencing very little visual motion or retinal slip. And we can repeatedly pair this for tens of minutes, just as we had done for opposite direction visual motion. And what you can observe in the animal's behavior is that there's a very little change in the gain of the VOR. And this is true across animals. So this is a minimally or non-adapting context that we just call gain stable for simplicity, because the gain doesn't change in this context. So the question is, what's happening to climbing fiber evoked activity in Purkinje cells in this context where the animal learns? Well, surprisingly, climbing fiber activity is actually deeply modulated as similar to this gain increase condition where we had delivered opposite direction visual motion. Notably, we did measure a greater amount of climbing fiber activity in this adaptive context, so that might contribute to differences. But certainly there's not a wholesale reduction in overall climbing fiber evoked activity. And so this got us thinking back to this original idea that we presented, that complex spikes are really outputs, and when the critical signals for plasticity induction often or would likely relate to the dendritic calcium response, because plasticity will be induced in a calcium-dependent manner. And therefore, it might be important to follow what's happening to the calcium response in Purkinje cells during these different contexts. And to do this, we turn to fiber photometry, and just notably, recording in the flocculus is really difficult to do it optically, because the flocculus is buried sort of in the inner ear. And doing that with 2-photon microscopy is rather challenging. For example, turning a microscope is quite hard to do, in addition to just accessing that structure. And so in this method, we transduce Purkinje cells with GCamp, here GCamp6F, and then implant an optical fiber to target the flocculus, deliver light to excite GCamp, and then collect the emitted fluorescence back through the optical fiber. We previously showed you can record large transients that likely relate to climbing fiber evoked activity. And then what we could do is stimulate the animal in these two contexts, this adapting context that would generate a learned increase in the VOR, as well as this minimally adaptive context where we just have a stable visual surround. And notably, when we look at the average calcium response, there is a larger response in the adaptive stimulus rather than the non-adaptive stimulus. And this really strikes us as the critical neural correlate of learned motor behavior, rather than just modulation of complex spikes, which is output. It's really the input side that is the dendritic calcium signature that might give way to these learned changes in behavior that would relate to plasticity induction locally within the flocculus. So what contributes to these differences in calcium signatures within the Purkinje cell population in this adaptive stimulus? Well, we've had this longstanding interest in the potential role of molecular layer interneurons. This is an embedded GABA-releasing inhibitory interneuron, obviously by name. They reside in the molecular layer. MLIs receive excitation from parallel fibers and can drive a profound feed-forward inhibition onto Purkinje cell dendrites, where the climbing fiber synapses are located. We performed a set of experiments several years back that shows the potency of MLI-mediated inhibition to regulate the climbing fiber evoked calcium response. I'll share that data with you. Here we made recordings in acute brain slices from cerebellum, filling the Purkinje cell with a calcium dye, Flo5F, and then using 2-photon microscopy to measure the evoked calcium signature to electrical climbing fiber stimulation. You can see the evoked calcium response here below, simultaneous with the somatic complex spike that we're recording simultaneously in the soma. But in this set of experiments, we also expressed the red light sensitive channel rhodopsin variant breaches in MLIs so that we could photostimulate MLIs concurrent with the climbing fiber stimulus. And when we did this, you could see a robust reduction in the size of the calcium response in the dendrite. However, the complex spike in the soma was relatively unchanged. So this just emphasizes the potency of MLI-mediated inhibition to regulate the dendritic calcium response, and also to show you how uncoupled the dendritic calcium signature is from the complex spike at the soma. So to address whether MLIs are having a potent effect on on dendritic calcium signaling in Purkinje cells during the VOR, we sought to manipulate their activity while simultaneously measuring calcium signaling in that Purkinje cell population. We did this by using this complicated targeting scheme, but to summarize, we basically expressed GCAMP in Purkinje cells and halorhodopsin in MLIs so that we could measure the Purkinje cell calcium activity within their population, either in the intact condition or after we disinhibit it by photosuppressing MLIs during the performance of the VOR, and measuring this using photometry. And so this is the net result from an example mouse. And black is the calcium signature that we record during the VOR in the control condition to this minimally adapting stimulus. This stimulus where there is a non-moving visual surround. However, in trials where we photosuppress MLI activity, you can see increases in the calcium signature within the PC population, as shown here across mice. So this indicates that we can suppress MLI activity, and the net result is an increase in the calcium activity within the PC population, indicating that that activity is under the regulation of MLI-mediated inhibition in this context. Notably, we also observe what's happening to the VOR when we acutely suppress this MLI activity in this context. And this is the result of that. If you follow the eye movement to the acute photosuppression of MLIs, there's actually no net effect. That is, the VOR, it remains unchanged. So this this increase in calcium activity is not due to inadvertent error that optogenetic MLI activity would produce. Rather, it's a local effect emergent from the circuitry from Purkinje cell disinhibition. That is, in the absence of MLI activity, calcium responses get much larger in this particular region. We were curious to know what would happen if, instead of just acutely suppressing MLI activity, rather we suppress the activity across multiple trials, as if the animal was receiving visual feedback errors, as if it was making a mistake. That is, if there were, similar to our training procedure where we introduced opposite direction visual motion. Oh, sorry, I apologize. This is, yeah, so this is in this condition where it is minimally adaptive. We could perform control sessions and show that, again, animals don't learn in this condition, or the VOR is unchanged. However, we can then retrain the animals days later, in this case, photosuppressing MLIs in a directionally biased way, either during the ipsiversive or contraversive phase of head stimulation or head turns. And measuring what the net effect of that is by measuring the VOR isolated in darkness before and after repeated pairing. And what you observe is that the animals learn to alter their VOR in this condition. Specifically, they increase the gain of the VOR as if they were experiencing a motor error. But as I showed you, with the acute suppression, there was no net effect on on the VOR through acute stimulation. So this to us, this results from the chronic or the consistent elevation in Purkinje cell dendritic calcium signaling during that stimulus. I'll also note that the VOR, that this photosuppression of ML activity either during the ipsiversive or contraversive phase was equally potent at resulting in a learned change in the gain of the VOR. Notably, we also, within the Purkinje cell population, we observed climbing fiber evoke activity on both phases, indicating that either one can become potent for generating learned changes. Also note that this was an animal that already had a well-calibrated response, so these learned changes are maladaptive. So our interpretation of these results is that in contexts where the animal shouldn't learn, MLI activity diminishes the potency of climbing fiber evoked calcium activity, and that prevents plasticity induction that would lead to alterations in the VOR. So the next question was what would happen to VOR performance if we photosuppressed MLI activity in this adapting context. And so this is an illustration of the procedure where we could introduce opposite direction visual motion. Again, this context that will lead to an adaptive increase in the gain of the VOR, and then photosuppress MLI activity. Here we did this on the ipsiversive phase of vestibular stimulation. And the net effect of this was nothing. That is, the animals learned similar to the control condition. So we did not interrupt the animal's ability to acquire an adaptive alteration to counter this sensory feedback that indicates an underperforming VOR. So one interpretation of this result is that there's no effect on this system because the Purkinje cells are already disinhibited. That is, MLI activity is already reduced in this context, leading to elevations in calcium that are sufficient for plasticity induction that would alter the mouse's performance. We also did this experiment on the contraversive phase of vestibular stimulation. Notably, during VOR adaptation that I showed you, it is believed that the climbing fiber signaling on the ipsiversive phase is the activity that will drive these learned changes. When we photosuppressed MLI activity on the contraversive phase, there was actually a strikingly different result. Rather than no effect on learning, we actually observed the absence of learning in this context. This was at first somewhat head-scratching to us of why we saw this difference. However, we sort of thought about this for a bit and wondered whether it has to do with the directionality of VOR learning. That is, there are muscles that pull the eye in each direction during the VOR. And you want to strengthen one direction as opposed to the other in response to visual motion. And therefore, we hypothesized that there might be a regulatory process that diminishes the ability to generate learning on both phases of motion. Else, if both phases of vestibular stimuli were equally potent at producing learned changes, you would actually produce an isometric effect. That is, would counterbalance by pulling in each direction of the eye movement. And to explore this in more detail, we wanted to ask what the response, the potency of the climbing fibers to produce calcium activity was on each phase of the vestibular stimulus. To do this, we transduced climbing fibers with this red light sensitive breeches and then expressed GCamp in floccular Purkinje cells and then measured the optic genetically evoked calcium response in those PCs to that stimulus. Here's an example in a quiescent mouse where we photostimulate. We can produce large elevations in calcium that are consistent with engagement of the postsynaptic response to that input. However, for this set of experiments, we photostimulated climbing fibers with each phase of vestibular stimulation. Here we did this on the ipsiversive phase of vestibular stimuli, and this was highly effective at generating large increases in calcium. But notably, when we stimulated climbing fibers on the contraversive phase, it actually had a very weak effect on promoting additional calcium drive. And so as I previously mentioned, we believe that large elevations or increases in calcium signaling within the PC population is going to be a signature for learned changes. And therefore, if we monitored what happened to the VUR performance by repeatedly pairing this climbing fiber stimulus, either in the ipsiversive or contraversive phase of vestibular stimuli, we generated strikingly different results on performance. That is, the ipsiversive stimulation is highly effective at driving a learned increase in the gain of the VUR. However, climbing fiber stimulation on the contraversive phase is rather impotent, it isn't very effective at producing learning. And we think that this largely reflects the ability of those climbing fibers to mobilize Purkinje cell calcium signaling that would be sufficient for plasticity induction and learning. So we're sort of thinking back to this idea, what would potentially regulate the ability of these climbing fibers to produce learning in these different contexts. Again, our belief, or at least the idea, was that it was due to regulation by molecular layer inhibition, and molecular layer inhibition is not only regulating in the particular context, but ensuring that climbing fibers are directionally biased to promote learning in a single direction. To explore this, we turned to dual color optogenetics, where we transduced climbing fibers with red light sensitive breaches, and also expressed blue light sensitive GTCR2, which is an inhibitory opsin in molecular layer interneurons, and then revisited this experiment that I introduced by photostimulating climbing fibers during the contraversive phase of vestibular stimulation, which is normally impotent at producing learned changes. So that's shown here in this control experiment with climbing fiber only stimulation. However, if we pair that climbing fiber stimulation with MLI photosuppression, we can generate a learned change. So we could uncover the latent ability of climbing fibers to produce that learning when we reduce the activity of MLIs. And this wasn't due to suppressing MLI activity alone, because in trials with just MLI activity suppression, we cannot recapitulate this learned change. So this suggests that for learning, for climbing fiber induced learning, that the system needs to overcome MLI mediated regulation of Purkinje cell dendritic calcium signaling. And so we were thinking about ways that the local circuits could produce disinhibition of Purkinje cells. And we knew from longstanding evidence that MLIs are synaptically coupled, that is, they inhibit one another, and therefore inhibition between MLIs could promote Purkinje cell disinhibition. And therefore, the activity between MLIs, the inhibitory activity between MLIs, might be the underlying mechanism which leads to enhanced dendritic calcium signaling through disinhibition. That is, that it's an active process in contexts that require learning. To test this hypothesis, we sought to remove inhibition between MLIs, and we did this through this molecular genetic approach, using a tool that Don Arnold's lab at USC developed. This is called GFE3. What this is an E3 ligase that's directed to the GABA receptor scaffolding protein gefrin. It's genetically encoded, so we could deliver it specifically in MLIs, and gefrin's expressed postsynaptically, so this is just a postsynaptic effect. We could express GFE3 in molecular layer interneurons. This is an example recording from a slice from a GFE3 expressing MLI, or recording inhibitory activity, and you can see there's very little response relative to control injected animals, and this is the net effect across all recordings, where essentially inhibition is absent between MLIs. Importantly, as I mentioned, this is a postsynaptic effect, so we could also record from Purkinje cells which aren't expressing GFE3 and measure inhibitory activity, and there's actually no change. Again, that's simply because GFE3 is only in MLIs and not in Purkinje cells. So to summarize this simply, we removed inhibition between MLIs, so we prevented disinhibition of Purkinje cells. That is, MLIs continue to inhibit Purkinje cells, but they can't inhibit one another. So we wanted to know what the effect of this was on the calcium signal on the PC population. Again, we could turn to photometry by measuring the calcium response in the flocculus in GFE3 or control injected animals. As I previously introduced, when we introduce opposite direction visual motion, we can record an increase in the calcium signature of the Purkinje cell population, that is in control animals. However, in GFE3 animals, we see a strikingly different result. That is, despite the animals are experiencing opposite direction visual motion, which would be interpreted as an error, we see no change in the Purkinje cell calcium in the calcium signature in the PC population. And provided that we think that the increase in calcium is a neurosignature for learning, we wanted to address what happens to the VOR in animals that cannot, where Purkinje cells cannot become disinhibited, and where these calcium responses are greatly diminished, with the hypothesis that this should prevent learning. So in control injected animals, we could subject them to opposite direction visual motion. And as I've repeatedly shown you, this is very efficient at producing a learned increase in the gain of the VOR. However, GFE3 injected animals, there was a strikingly different result. That is, the animals could not adaptively increase the gain of the VOR to this opposite direction visual motion. So despite the fact that they experienced a sensory feedback, that they were generating an error, they were not adaptively regulating their response. And this was true across animals. So Purkinje cell disinhibition seems to be critical then for instantiating that learned change. So by what mechanism might explain this? This begs the question of what is the logic of MLI organization. We know from work dating back with Ramon Ika Hall that MLIs come in two potential subtypes based on morphological differences. There's a soma targeting basket cell type and a dendrite targeting stellate cell type. Again, provided this was based on morphology, Adam Hantman and Vilas Menon, two collaborators, and I wanted to explore what was the genetic diversity of MLIs to ask what their potential diversity is. We did single cell RNA sequencing from fluorescently labeled MLIs and looked at the landscape of genetic diversity. And rather than seeing one cluster that might represent the continuum of the same class, or two clusters that might represent stellate cells and basket cells, we saw something like 3 clusters. We identified genes that can demarcate these, and then used a small molecule FISH to label populations from these, and we could clearly see differences in gene expression within different MLIs. And certainly when we looked at where these MLIs exist, we could see differences in their general location with, for example, NFM mark cells existing more deep in the molecular layer and SP5 mark cells more superficial. And there's this third class that is represented throughout the molecular layer. Subsequent to this, or more recently, a work from Wade-Reger and Sun Lab in South Korea have exhaustively looked at this genetic diversity in their connectome. And this new picture of MLIs have emerged rather than existing just as basket cells and stellate cells. There's something more like two general classes, MLI1 and MLI2. MLI1s can be subdivided into MLI11 and 12. Likely, these assort to what you would classically know as basket cells and stellate cells, although the differentiation between those is probably relatively weak. They might be the continuum of a single class of Purkinje cell targeting cell, and notably MLI2 seems not to prefer Purkinje cells, but rather inhibit other MLIs that would disinhibit Purkinje cells, similar to the function of VIP-marked interneurons in forebrain that generate pyramidal cell disinhibition, have been shown to be potent regulators of plasticity and learning in those systems. And so one idea is that MLI2 might be a master regulator of such activity and provides a neurocircuit explanation of why you would have differential engagement of the MLI microcircuitry in these different contexts. So again, what we find is that MLIs gate climbing fiber induced activity. They prevent large elevations in calcium, which would be super potent for plasticity induction like LTD, and eliminate non-productive alterations in behavior. We think that there's an active mechanism through Purkinje cell disinhibition that leads to large increases in calcium, sufficient for LTD induction, unlocking the gate for learning in these erroneous contexts. We think that this is necessary because unregulated plasticity is potentially dangerous, as again I introduced. And without constraint, you could generate maladaptive behavior. We know that Purkinje cells have a limited number of synapses, and therefore if they continuously underwent plasticity, you could generate saturation of those, just like if you took a lot of pictures on your cell phone, at least it's not hooked up to the iCloud, and prevent additional memory capacity within that circuit. So we were interested in examining this. As I showed you, or as I introduced earlier, we know that perturbations to acute perturbations to MLI inhibition do not produce alterations in the VOR. However, we sought to do a chronic manipulation, and for this, we turned again to GFE3 and expressed that in Purkinje cells. So now Purkinje cells were chronically disinhibited. And this is again as an example of recording from a slice showing that GFE3 expressing Purkinje cells don't have much inhibition. And then we could measure what happens to the animal's VOR across time. Here's a snapshot quite late in this, after weeks of GFE3 expression, and note that the gain of the VOR has increased over time as if the animals had experienced errors. And second, we could subject that animal to opposite direction visual motion to discern how it was adapting to that stimulus. And rather than increasing the gain of the VOR, which control injected animals did, the GFE3 injected animals that were chronically disinhibited did learn, but they learned in the opposite direction. This was a maladaptive change, so they were exacerbating and or poorly performing VOR. So this is unproductive. We did a similar manipulation. Rather than doing GFE3, we did chemogenetics to chronically disinhibit Purkinje cells and observe for corresponding changes in the ability of climbing fibers to produce plasticity rather than producing LTD. We see instead LTP, which this sort of opposite direction sensitivity often results from saturation of one type plasticity. That's generally true across plasticity systems, and opposite direction plasticity might explain why you why the animals experienced opposite direction learning. So we've sort of been exploring this in more detail and out of time, but we think that unregulated plasticity as potentially a disease state that is without regulation, you could accumulate maladaptive responses that could explain a slew of cerebellar dependent abnormalities. And we can use the VOR to explore these, looking for common phenotypes, both in mouse models as well as human patients, looking for translated responses. And we're actually seeing this in numerous disease states. So we're quite interested in exploring this in detail, not only for mechanistic insight and therapeutic and diagnosis, but potentially developing therapeutic agents that could help ameliorate symptoms associated with that. So finally, I just want to thank people in the lab, acknowledging this was work largely of a graduate student in the lab, Ka Zhang. Zhen Yang, a postdoc in the lab, has taken over a number of these studies with the help of Abid Nashif and Abby Person's lab, who's here with DiGregorio and I. We've have a sort of super group focused on cerebellar function, and also just acknowledging the importance of collaborators here with Adam Hantman and Vilas Menon, but also Don Arnold, who provided this GFE3 agent. And with that, I'll open it up for questions.
Thank you so much, Jason. Gosh, that was absolutely stunning. There were so many beautiful new things for me, certainly. So let's open it up for questions. Please just raise your hands if you'd like to ask Jason some questions. Maybe I can start. Jason, can you go back to the results that you showed with the climbing fiber activity electrophysiologically measured during stable condition, and you showed that there was that surprising result that there was lots of modulation of the climbing fibers. Can you just teach us a little bit about the difference between what this is measuring and what the calcium response is measuring using optical imaging?
Sure. Yeah, so this measurement, so this is electrophysiology. We're finding individual Purkinje cells and again, measuring the rate of complex spiking here to a vestibular stimulus. What I'm showing you in gray is the head position. So as the head is being moved back and forth on watching this modulation. In the photometry, we're expressing GCamp in Purkinje cells and measuring calcium activity within that population. So there's a couple caveats there. This is captured across the Purkinje cell population, so that's not individual Purkinje cells. In addition, the calcium signature the activity that's producing that is not fully explored based on a lot of two-photon imaging that we've performed, as well as many others. We know the dominant calcium signature is, sorry, I don't know why this is popping up. Let's stop this. There we go. Oh, let's keep popping up. Okay, so what is producing that calcium activity is likely to be climbing fibers, but probably not exclusively. And so that's an important caveat to understand, and so why I'm trying to characterize that as the PC calcium response. We'd love to turn to recordings from individual cells. Certainly we've done that in a different system. I didn't have time to talk about that. where we disinhibited the lateral cerebellum during a different movement task and showed at the level of individual Purkinje cells that there was a robust increase in the size of climbing fiber evoked calcium responses in the disinhibited condition. So we know that result holds true at the level of individual cells, but we'd love to do that eventually in the flocculus. Just doing that is not as straightforward as it sounds.
Hey, Jason, can I just follow up on that really fast?
Sure.
Have you looked with the silicon probes to see if there's any distinction between the fat spikes you see in the molecular layer and the complex spikes that you're recording somatically that would sort of match this distinction you're seeing between the calcium imaging and the complex spikes?
Yeah, that's a really great question, Court. The short answer is no, we have not. And so just to give, so one idea out there is that on these silicon probes, you can pick off not only the somatic response, the complex spike, but there's a specific neural signature in the dendrite that correlates with that climbing fiber response, and these have been called fat spikes. Whether those fat spikes are truly climbing fiber evoked, also completely unknown. Though they can be identified and followed, it would be totally worthwhile to do, and we just haven't done that. And second, I just don't know how sensitive the duration of that fat spike might be with the correlation in calcium. Again, it's an approachable question, great idea, just limited times on our hands, and you know, my preference would be to utilize our time to attempt to measure those calcium responses directly, because I think that's the true correlate.
May I ask you a question?
Sure.
Thank you. The talk was amazing. So I was wondering if you looked at the MLI's activity during the adaptation. Basically, the question is that if they are mediating learning, how are they learning? How are they changing to know how to mediate the learning? What is the mechanism that you think is?
Yeah, so I mean, we would, one hypothesis is that the different MLI populations are engaged differently depending on those contexts. We're actually performing those experiments now. So I wanted to highlight what Jen and Abed have been doing. But the data is a little premature to show at this time point. That's the way to go about it, and that's what we hypothesize would happen. But again, that's to be determined.
Jason, David Linden here. That was really great. I enjoyed your talk. So is there any chance that there are functional GABA receptors on the climbing fiber terminals and that the molecular layer interneurons might be engaging them?
So yeah, could the MLIs be inhibiting presynaptic GABA receptors? That's an interesting question. I actually don't know if climbing fibers have GABA receptors. I will note, I can show this with you. This is a preprint that people might be interested in. I highly suggest they look into it. Again, this is going back to data that the Sun Lab have generated in Seoul. Where is this? This basically is just going to quickly summarize their net result. Here. So what they found in their connectome is that MLI2 is getting a direct input from the climbing fiber. And so climbing fiber activity signaling to MLI2 might actually produce this inhibition of the Purkinje cell. You could imagine in sort of gradient that to repeated climbing fiber stimuli that there might be then an active mechanism on subsequent climbing fiber induced responses, that there's disinhibition. So basically the MLI2 would have access to what the climbing fiber is doing through a direct excitatory connection, which would produce a different inhibitory state, for example, disinhibition. So that's what I would surmise would be the critical way of delivering an input that would change how the neurocircuit is response to climbing fiber stimulation. Again, this is pretty early. Again, this is coming off of a preprint, but I'm sort of fascinated by the potential implications of that and what it's driving in the response. And again, I can't directly address the GABA question, probably reasonable. There are a number of other neuromodulators that it could work through, endocannabinoid signaling or something similar. Sasha, you had a question?
Yeah. So to comment on a question, first, Jason, I think I've said this to you before, but I'll just say here in public, you are the best thing that has happened to the vestibulocular reflex field since it was founded, other than our work, of course. But
wow, thank you, Sasha. That's coming from you. That's such an honor.
It's spectacular, yeah. And so the question is this all for gain increase learning? So to distinguish it from gain decrease learning, two things kind of really super relevant. One is clearly a cerebellar cortical phenomenon for the most part. And the second is short lasting because it's energetically more demanding, more spikes. In general, making more of something takes more energy than making less of something. Yeah. So so how are you thinking about the other direction, or are you just like, it doesn't matter because there's all sorts of cerebellar learning that is working using this kind of scenario?
Yeah, so we did all of the gain decrease training. So this is same direction visual motion. And for the non-aficionados, this gain decrease learning is believed to not depend on climbing fibers. So this should be climbing fiber independent learning. And the net effect is that all these manipulations all this disinhibition doesn't have any effect on that. So we did all of those experiments. Gain decrease learning is unaffected by this. So we think it's specific for climbing fiber evoked learning. I don't know if you also asked if the sort of what the lasting effect of this is. We didn't follow that over time. I think that the effect of the chronic PC disinhibition begins to speak to that. I think that would produce a lasting effect. But I think in terms of the duration of learning, we know that learning exists on different timescales. And there are different mechanisms that could potentially explain this, and disambiguating what those what the roles are on different timescales of learning would be interesting, but we just haven't pursued that.
Great. And just a little final question. Are there, like in your recording, sort of sense multiple flavors of Purkinje cells that are distinguishable in the flocculus other than the directionality?
Yeah, I'd love to know that.
Okay.
But
nothing jumps out at you though from your recordings.
We have not done that. I mean, I think that is, that's just not going to happen in flocculus. It's going to happen everywhere. That we want to know the neuro, like right. Whether function follows different types. We know that there are different subtypes of Purkinje cells, and whether they are engaged differently and have different signatures is an open question. I think that would just be fantastic to follow up on.
Great, thank you. And thank you again, Reza, for hosting this.
Oh, my pleasure. Gosh, what a great way to learn. That was amazing. Any other questions for Jason?
Dr. Christie, I had a small question, if you don't mind.
Sure.
If you can go back to the slide that Reza referenced initially, showing the complex spike response from the phys recordings.
Sure. Yeah. What do you want to know?
Yeah, so I noted that, so first of all, were the traces you were showing the average across multiple Purkinje cells
Yeah.
complex?
That was everything. That was everything across, that was averages of everything. So across Purkinje cells and across trials.
Okay, so I noticed that there was a positive peak in the complex spike response, probably a shift up from the baseline of the cell. And then shortly before you rotated the head in the other direction, before that trough occurred, there was a smaller same direction. Does that reflect some variability in the preference of the complex spikes response and the timing of it with respect to the phase of the movement of the head? Like, do different cells care about a different point of the
Yeah, they do.
head?
So we could show you Purkinje cells that follow that are tuned to each direction. Usually the complex spikes typically align, but not exclusively, to the peak phase of head velocity, which would be the point where the retinal slip would be the greatest, but it's not. It wouldn't be exclusive. So that this kind of speaks to what Sasha was bringing up. Does this relate to different types, subtypes of Purkinje cells? What is the diversity out there, and what are they tuned to, and what their role is in this particular behavior? I would venture to bet that there is going to be a diversity, and that diversity is important for something, and that we might be sort of scratching the surface of that. We're getting access now to a greater number of Purkinje cells with changing our recording configuration. And so it'll be really interesting to explore what happens when we have a very large population to look at.
That's amazing. Thank you.
Ramki, you have your hand up.
Yes. Thank you so much. It was a very interesting talk. And I'm relatively new. I'm unfamiliar with this vivo or learning and vivo related thing. I just one question. Just wondering. Also, missed a few earlier slides. I'm sorry about it. So, if I understood correctly, the molecular layer interneuron activity is suppressed due to that. It actually enhances disinhibition of Purkinje cell activity. So that eventually enhances the climbing fiber induced learning. Is that correct?
Did the climbing fibers induce learning?
Yeah. So my question is, so under natural condition, so what actually causes the suppression of molecular layer interneuron activity, which would eventually disinhibit the Purkinje cell?
Yeah.
If I understood. Yeah.
Yeah, we don't know. What I introduced is that potentially the climbing fiber itself might be doing it. So again, this MLI2 class of MLI seems to have direct climbing fiber input, so it has access to that message. Whether it's similar or different than the Purkinje cell is an open question. For example, you could have differences in short-term plasticity that dramatically change the landscape of how that activity is being engaged. Second, there is slice data that indicates that climbing fibers can communicate to MLIs by spillover-mediated transmission. Whether that holds true in vivo, I think, is now under active debate. In addition, MLIs receive parallel fiber activity, and it might be that the channels of mossy fibers that are engaging these systems also are delivering information differently, and so that would be a potential way to change that, so that mossy fibers and granule cells might be tuned to different aspects of the behavior. For example, visual motion versus motor output, and so I think that has not been pursued in a lot of detail, and so unfortunately that. Addressing your question of what is the mechanism that would produce a potential differential engagement of these populations for and produce Purkinje cell disinhibition is not currently known. Yeah.
Thank you.
So I will. I don't know if there's any more questions. provide this as a great group of people that might be doing similar things. One thing to keep track of is if you're looking for changes in perhaps disinhibition through Purkinje cell simple spiking, keep in mind that the Purkinje cell dendrite is highly electrotonically isolated. So inhibition that you're going to see is probably very biased to the soma, and you're not picking off what would probably be happening in the dendrite very well. So just be careful if you're conducting a similar analysis on Purkinje cell simple spiking. It might not be clearly informative of what's happening in the dendrite. Just planting that seed out there.
Jason, that was amazing. Thank you so much. Thank you, everyone, for the wonderful questions, and what a pleasure to be with you on this Tuesday. Have a great day.
Thanks again. Talk to you later.